So today I'm going to talk the work of this PhD student, Francesca Sacco, whom I convinced a couple of years ago when she started a PhD to try to give some contribution on a very simple but important problem. So let's suppose that we have one of these cartoon pathway that we have seen during over the past 2 days. Let's say a receptor phosphorylates C that then inhibits D, and then we have some function at the end modulated by this effector. And so now we want to know if any of the other 25,000 gene is in some way able to modulate this pathway or participate in its function. So the question we would like to address is protein X linked to the pathways involved in modulating the activity. And second, how precisely can we place it in a pathway graph? So can we only say that X some way affect the pathway, or rather we can say that X dephosphorylate B or phosphorylate E? And can we use a strategy that can be applied in a relatively large scale? So not one protein, one pathway, but many protein, many pathways. So since we have some support from Italian Association on Cancer Research, we focused on a series of pathways here drawn in a very incomplete way as linear pathways that are involved in cell growth or homeostasis. So the pathway induced by cytokine ending up in P38 activation and FKB migration to the nucleus, or receptor tyrosine kinase. But these are not all the pathways that are involved in cell homeostasis. SMAD pathway is not here, STAT pathway is not here. But clearly, as we know, there are many cancer that are which we find mutation in most of these highlighted proteins. So this is are very important pathway in the modulation of cell growth. So the question, again, in this slightly more complicated pathway, we have protein X. Is this protein X affecting the readout of these pathways? So, as you know, most of these green lines or red lines are actually activity of some enzymes that either add or remove phospho groups from the proteins. And this, by a variety of mechanisms, activates or inactivates the protein that is downstream. And protein phosphorylation are mediated by a large family of enzymes called kinases. And while conversely, the reverse activity that removes the phosphate from the protein are governed by another large family of enzymes called phosphatases. So for some reason, kinases have been much more popular than phosphatases over the past 10, 20 years. And kinases are considered the sexy enzyme that regulate the pathway, while phosphatases are more or less housekeeping enzyme. So if you look at in PubMed, and you look at abstract that cite kinases, you see the steady increase of paper describing kinases, while the number of papers that describe work on phosphatase is much lower. And this becomes more dramatic if instead of counting the paper, you do the total impact factor. So clearly, kinase papers tend to be published in higher impact factor journal than phosphatase journal. So if I have to give an advice on young people here, if you want to do a productive PhD with a lot of paper on high impact factor, don't work on phosphatases. Concentrate on kinases. But fortunately, I was able to convince Francesca that things might change in the next few years, and so it was worth investing on the phosphatase family. So the human phosphatase family is a very large family. Here I have an evolutionary tree of approximately 300 proteins. These are phosphatase in a broad sense. So in this tree, I also included regulatory subunits that regulate the activity of the phosphatase or direct the phosphatase to their substrate. Then there is a relatively large group of tyrosine phosphatases, some specific for tyrosine, some other that are dual specificity, so they phosphorate both tyrosine and serine threonine, and so on and forth. Here I've also included the nucleotide phosphatases and lipid phosphatases, and a number of proteins that by homology look like phosphatases, but nobody knows whether they are really phosphatases. So these are the proteins that Francesca decided to study. So, and in order to approach it in high throughput fashion, she used this technology that has been developed over the past few years, where you have a collection of a small interfering RNA that you print on glass slides together with transfection reagent. Then you grow your cell model of choice on the glass slide, and where you have spotted the cRNA with the transfection reagent, you get transfection of your cells, and then if the cRNA is changing in some way a pathway in the cell, you can measure it by the use of specific fluorescent reagents. And you do it not going one spot after the other, but you do it with an automatic fluorescent microscope that in a few hours can identify all the spots in which the phenotype has been changed. Actually, it can measure the phenotype. Of course, cRNA can have off-target effect, so to minimize this, we use 3 different cRNA per gene, and to work on reproducibility, we use 3 different replicas. At the end of the day, you have approximately 100,000 images that you don't want to look on the computer one after the other. So you use automatic image analysis software that are open source, easily available, that don't cost anything, and at least the one we are using is very effective. And so you measure, most of the time by measuring fluorescence, you measure the activity of key proteins in the cell. So if we go back to our very simple pathway or series of linear pathways, we have defined a number of what we call sentinel proteins that have been assigned for their centrality in the graph and for the availability of robust readouts that you can use in a high throughput fashion by this technology. And so we have shown activation of P38, ERK, NFKB, the formation of LC3 dots to monitor autophagy, and so on and so forth. So for instance, this is what you observe when you look at the phosphorylation of S6. In the absence of nutrient, you see hardly anything. If you put nutrients, then you get activation of X6. And this is an equivalent activation of P38 by addition of TNF-alpha. And these are the occurrence of LC3 dots in absence, in starvation. So now we can look at all our images, and we try to see how the interference by one after the other of the 300 phosphatases affect these 7 phenotypes that we have considered. So if you now plot in the measurement, here you have the intensity, for instance, of S6 phosphorylation, and then you see how many phosphatases have the value corresponding to the averages. Most of these have a value corresponding to the average, and so on for the LC3 dots. But then if you go into the tail of this quasi-Gaussian curve, you see that if this is the control in which you see partially some S6 phosphorylation, this phosphatase completely, inhibition of this phosphatase eliminates S6 phosphorylation, while inhibition of this other phosphatase increases phosphorylation. So you find the tail of this curve, some signal in your experiment. And in a similar way for LC3, if you, perhaps you don't see here, but in conditions we are using, you should see some spots, even in the control, but these go to lower level if we use the phosphatase, if we interfere the phosphatase PTN21, while it increases with this other phosphatase. So by doing this repeatedly for all our phenotypes, and by choosing appropriate threshold, so statistical threshold, we can convince ourselves that some of these phosphatases are involved in the modulation of these readouts. So we can place them, you can place this list near the sentinel protein or the readout that are affected by the phosphatases themselves. And so the first thing that when you do this high throughput experiment, the first thing that comes to your mind that you're just collecting rubbish, that there is no real biological signal. So before going on and investing the time of under PhD student to study one after the other the relevance of this phosphatase in the pathway, you want to have to analyze them together and try to understand that you're doing things right, there is some signal. So the first thing that we did, so we did some validation. We did validation by repeating the experiment with independent siRNA library, not the one that we have used for the primary screening. We use independent experimental setup. For instance, for most of this validation, we now use Western instead of imaging. And then instead of only inhibiting phosphatases, we also did phosphatase overexpression, where we expect to observe the opposite signal. And by doing this, we observe that 70% of the hits are confirmed. So now, if these phosphatases are involved in some way in cell growth homeostasis, we suspect that in some way the cell cycle timing should be changed. And so we measure, by measuring the number of cells that are in mitosis after overexpression of these phosphatases. And what we observe that while the phosphatases that are positive regulators tend to, that have been identified by our assay as positive regulator, tend to increase the number of cells that are in S phase, in mitosis. The ones that have been identified as negative regulators actually have a lower number of cells in mitosis. So another type of analysis that he did is to try to see how many phosphatases that have been already validated in the literature as phosphatase as an oncogene or oncosuppressor do we recover in our assay. So there are, as I said, we are analyzing 300 phosphatases. 107 of these are tyrosine phosphatases that have been recently analyzed in a review. And the group of Michel Tremblay reported that 35 of these 107 protein phosphatases have in some way been associated to cancer as an oncogene or oncosuppressor. So among our total hits, I call hits the phosphatate in some way, whose inhibition in some way affects our phenotype. So we have 57 phosphatases, serine phosphatase, regular subunit protein tyrosine phosphatase. 36 of these hits are protein tyrosine phosphate, so we can compare it with this data set. The overlap between the 35 protein tyrosine phosphate involved in cancer and the 36 hits is 17. So if we had done it at random by just choosing for a collection of 107 phosphatases our hits, the probability of achieving such an overlap is it's in the order of 1 per 1,000. So in some way, we are recovering from this screening some biological signal. So this is another way to represent our results. So as you see, the activity of sentinel proteins, phosphorate activated S6, LC3, and so on, are represented as nodes in the graph. And the phosphatases are represented as nodes, the phosphatases that affect this phenotype are represented as nodes linked to the phenotype nodes. In red are the link that suggests that the phosphatase is inhibiting this phenotype, while in green is the evidence that we have evidence that this phosphatase is activating this phenotype. And the intensity of the bonds depends whether this information was extracted by 3 scRNA, 2 scRNA, a little bit less intense, and this one was one out of 3 scRNA that provided this information. So, but I want to show this representation because I want to stress that many phosphatases do not only affect one phenotype, but many of them affect classes of phenotypes. So either these phosphatases have multiple substrates in the phenotypes that we are considering, or more likely, or together, this representation is too simple. And actually, as we all know, this pathway talk to each other. This is a crosstalk. And then it's possible that by affecting one single node, you actually affect multiple readout in our screening. And so how can we approach this? So since our analysis is not looking at a single readout, but it's a multi-parametric readout, so we can try this type of reasoning. So let's suppose that we have a phosphatase like P1 that affects readout of LC3 ERK, but does not affect S6. So if you look at this even simpler representation of the RAF pathway and this interconnection with the PI3 AKT pathway, so you can convince relatively easily yourself that this phosphatase must in some way interfere with these two nodes here. Because if it was interfering with PI3 kinase or AKT, should have also changed the activity of S6. And in a similar way, if we have a second phosphatase that is only affecting LC3 but not ERK, then this means that this phosphatase is affecting these two nodes here, and so on and so forth. So essentially, we can establish a relationship between the phosphatases and the node of the network, epistatic relationships. So everybody can do this type of reasoning on this simple network, but then if we go to the real network, we need some help from computation, because even Francesca cannot place phosphatase in networks as complex as this. So in our approach, what we did first is look at the literature, use our biological wisdom, and talk to friends, databases, and assemble a network representing what we think is relevant for the type of things that we are studying. So for instance, you see that here P38 is phosphorylated, and this is mediated by the presence of TNF-alpha. So each single circle here represents a differential equation that is governed by a variety of parameters, too many, the vast majority of which we don't know much. And so what we have to do is some way fine-tune the model so that it can reproduce our experimental results. So we want to optimize the parameters in the model, but minimize the difference between predicted experimental results. So Francesca did a lot of experiment by perturbing the network with a kinase inhibitor or combination of kinase inhibitors, and then measuring the activity of our SENTINEL proteins. So now we have experimental results, and there are well-established mathematical models to optimize the parameters in the model so they can reproduce experimental results. So once we get to a point in which the model is sufficiently robust and we trust the prediction, then we can stop doing experiment and see what the model predicts and trust this. So why this digression about modeling? Sorry, this is another example. For instance, if you interfere with the activity of Tor, then the model would predict that you induce autophagy, but you decrease the phosphorylation of X6, while, for instance, P38 and ERK and FKB are likely not to be affected. So I want now to define what we define as a cell state. So in each experimental condition, we measure these 7 different parameters, and the combination of these 7 parameters, we call it the cell state. And we represent it as a vector in a 7-dimension space, where the coordinate of the vector are our experimental measurement. So since not many of you can reason in the 7-dimension, so here I simplified in 2 dimensions. Let's suppose that we have measured P-ERK activation P38. The combination of these 2 measurements define a cell state. So now this cell state can be reproduced by our model. So by inactivating each of these nodes, for instance, if I inactivate PA3 kinase, I didn't pay any attention to draw it well, but P38 and P-ERK will change, so the direction of the vector will change. If I instead do an in silico experiment in which inactivate IKK, I get a different result, and so on and so forth. But then we also our screening, so in which we have a 7 measurement for the cell state after inactivation of the phosphatase. Now my interest is to understand is this DEP1 phosphatase, which node of this model is interfering with them. So I compare my experimental cell state vector with the computed cell state vector, and I measure the vectorial difference, and I conclude that the computational cell state that is closer to my experimental cell state is pointing to the node that is a likely target of the phosphatase. So this now is automatized, so we can do it for many interference, and we can come up with a more detail. Of course, we do not place phosphatase on a single node, but on a bunch of nodes that have the same properties. So for instance, here you see these 3 phosphatases interfere either with MPK, TSC, or mTORC. These phosphatases in some way affect autophagy, but there are a lot of proteins involved there, and we don't know the exact target. So the next step would be to try to map a bit more precisely these phosphatases by identifying their substrate. And to do this, we have developed over the past couple of years an approach. So this summarizes what I've just said. For instance, let's say that we have found that the DEP1 phosphatase PTP1B affects the readout of ERK. But in our model, all this bunch of reactions are summarized as a single node. So I'd like to know, is DEP1 interfering with GAB, with GRAB, with ERK itself, and what's the difference between DEP1 and PTP1B? So this is a technology that we have developed. So we were interested in finding out interaction between proteins and the phosphotyrosine proteome. So what we developed is a chip containing all the phosphotyrosine peptide in the human proteome. I mean, all the ones that were known at the time we started the project. So that we have 20,000 phosphotyrosine peptides, and now we can probe these 13 mers centered on phosphorylated tyrosine with any protein that we think could interact with phosphotyrosine. So this is done in triplicate. So this is meant to illustrate the reproducibility of the experiment in the 3-replica array. So we tested each peptide with a domain or with a protein. We use a fluorescent antibody labeled with a fluorescent probe, and we come up with results similar to this, where the intensity of the signal, the color reflects the intensity of the signal. So we have used this type of approach to map the recognition specificity of the family of SH2 domain that, as you all know, recognize phosphotyrosine peptide. So essentially, we have these 20,000 peptides on the chip. We probe it with SH2 domain bridge 1. We see which one are the peptides that react, and by aligning them, we see that this SH2 domain likes to have a leucine in this position, while if we take a different domain, like the one at GRAB2, it likes to have an asparagine at position -2. By repeating this experiment for the 120 SH2 domain of the human proteome, we were able to group them into the specificity classes, 17 specificity classes, each characterized by different contexts of the phosphotyrosine, the central phosphotyrosine. Incidentally, if we now, this is a specificity tree. This is an evolutionary tree based on sequence homology. And then we have mapped on the SH2 the color corresponding to recognition specificity. And you may notice that recognition specificity does not match sequence homology. So the colors are spread everywhere. So sequence homology, in a broad sense, is not a predictor of recognition specificity of SH2 domain. So it would be nice to do it also with phosphatases, but phosphatases do not tend to interact very strongly with their substrate. It's just a transient interaction. But there is a trick that's been developed by the community in which you can manipulate the active site of phosphatase by calling what's called trapping mutant. So by changing one or two amino acids in the binding site, you maintain the recognition specificity of the enzymatic substrate, but you prevent to carry the reaction through. So you stabilize the interaction and you transform an enzymatic assay into a binding assay. So we did similar experiment, similar to what I've just described for SH2, and we could map 16, the specificity of the 16 of the protein tyrosine phosphatases. So, okay, thank you. So now we have some information about the substrate that each phosphatase likes, but this information has a very low information content. So we tend to overpredict because the specificity of tyrosine phosphatase is not very high. You can see it from this logo. There is no specific selection. So what we set out to demonstrate that actually enzymatic subset selection by phosphatase is mediated by this enzymatic specificity that I just described and by a sort of network distance. So in some way, the phosphatase is brought near its substrate by some protein-protein interaction, and then the phosphorylate decide that there's some matching with its preferred substrate. So we can use Bayesian integration of the two features. You see that the prediction, the ability to predict the substrate by using in green PSSM is actually this, a preference for enzymatic pocket. We get this curve. If we use instead network distance to predict, we get the blue curve. If we combine them by Bayesian integration, we get a better curve. So both things are important, and if we use them both, we get a good prediction. So the point is to identify the target of DEP1 and PTP1B in this pathway. And we use the technology that I've just described, combining enzymatic specificity and network distance. And we were able to prove that PTP1B acts on the ERK pathway by dephosphorylating the GAB1 protein, while DEP1 directly dephosphorates ERK1/2 kinases. This is published, as you see, so you can have a look. There are a lot of gels of this type to convince you that actually these are the phosphatases that dephosphorylate this target, and by doing so affect the ERK pathway. So in conclusion, we have, by using these two types of approaches that I've just described, we identified the target of PT1B in the ERK pathway and the target as well of DEP1 in the ERK pathway. So we have added two new modulators of this pathway. So I've tried to describe a strategy to place new gene and gene product into complex pathway by doing high content siRNA screening. And I've shown a peptide chip approach to map binding specificity on a proteome scale. So as I said, this is the work mostly of Francesca Sacco, although other people have helped here and there, Anita Palma, Michele Tinti, Aurelio Nardozza, Sereno Paluzzi. In some phases of the project, I want to thank Ian and Rainer Peppercock for help in training Francesca at the beginning of the project. And most of the measurements were done at high throughput microscopy facility at Mario Negri Sud with the help of Antonella Ragnini. Okay, thank you for your attention.
Questions?